Hello friends, in this colposcopy training module, today I'm going to tell you that why high-risk HPV DNA testing is not at all required. And you all know very well that I'm against the HPV vaccine. I will be giving some reasons for the HPV vaccine here and some in my HPV infection module. But here, basically, I'm going to talk about why high-risk HPV testing is not at all required. And we will be talking about the limitations, the district level reality, and then we will be talking about the vaccine endpoints, AHS adjuvants which are being used, adverse events, and long-term efficacy gaps. So friends, I'm the only one who tells you that not to give the vaccine and not to use or not to go for the high-risk HPV DNA testing. So first of all, we should understand our limitations, in which particular area or the geographical area in India we are living, what is the financial burden we are having, what type of the patients we are dealing with, and how much cost we are going to incur in giving the tests and then finally doing the treatment. So depending upon all these things, I repeatedly tell everyone that don't follow the guidelines because you are going to deal with the individual patients. And then guidelines are for the masses, they are not for the individual patients. We know very well that what is the cervical cancer burden because a lot of propaganda because of the vaccines is already there, and the infant mortality rate is much higher than the cervical cancer rate in our country or the incidence in our country. The number of the deaths also due to postpartum complications is also very high. For example, 6,25,000 children or infants die every year in our country. 40,000 women, they die because of the reproductive and postpartum complications. So the number is very, very stalling. It is very high stakes. But still, because of the pharma companies, they are pushing the agenda of the vaccine and high-risk HPV DNA testing. So we very well know that the incidence of cervical cancer is 1,23,907. Almost 77,000 women die in our country, and most of the time, that is 75% cases are in advanced stage, and we are giving the opportunistic screening only. The majority of Indian women who die of cervical cancer, they never had even a single pap smear, VIA test or HPV test in their lifetime. When I got my PG done way back in 1988, till 1995, we were taught and we understood that even if we do a pap smear once in a lifetime of a woman, we are going to decrease the chances of cervical cancer by 50. But now we are asking to go for the multiple tests, that is high-risk HPV DNA testing, then followed by co-test or going for the triage. But I think that was a very good advice given to us, and I particularly stuck to that advice. We also very well know that the organized quality screening with via PAP, LBC, and functional biopsy followed by functional colposcopy followed by biopsy and referral pathways is immediately actionable and affordable, unlike molecular HPV testing infrastructure. So we are going to see why this high-risk HPV DNA testing is much more costlier as compared to the pap smear or LBC. I'll be taking the example at the district level. So high-risk HPV molecular testing, it requires validated lab, it requires molecular lab with QC protocols. Transport is a problem. Time processing, temperature maintenance is to be there, and then IT system followed by the protocol. But if we see for the PAP or LBC cytology, we can do it, or even our staff or the ANMs can do it. If we and cytologist or the trained cytotechnologist can be provided with the microscope at the district or at the CHC level. And even if that is not available, then the via screening can be done by ourselves, by us, all of us. Definitely, it has been trained. Many staff in many states through WHO programs has been trained, and it has been clearly found that cervical cancer incidence can be brought down if we are going for the via also, and that is via screening is feasible at the community level. So, if we recommending primary high-risk HPV screening as national policy when district level infrastructure is absent, condemns millions of Indian women to no screening at all. The enemy of good is perfect. So, at least that is why this is one of the reason. I'm going to give you another reason. So, this is one that the infrastructure, and if you need to send the samples to other state or far off, then lot of other problems, not only the financial, lot of other transport problem, maintenance of the cold chain and all those is required for this, and definitely the test is costly. So the main reason for my reason of asking you people not to go for the high risk HPV testing is that I think all of you must be very well aware that all the cervical cancer is not HPV driven. There's HPV negative cervical cancer and there's HPV independent cancer. And they almost amount to 30% in India. So, high risk HPV testing cannot detect gastric type, clear cell, mesonephric, neuroendocrine. It cannot detect HPV-independent adenocarcinoma, and it has been found that even if you go for the high-risk HPV testing, it doesn't help in reducing the cervical cancer mortality. And triage or the co-testing is mostly needed, and that adds to the financial burden. So this is very interesting and very important paper which was published in International Journal of Radiation Oncology in 2022 by Dr. Jwala, which showed that India has got the 29% of the HPV negative cervical cancer at that particular place, and when after all technical false negatives were excluded, 21% remain HPV negative. So the global incidence of HPV negative is 5 to 11%, and India's HPV negative is almost three times. So that is 21 to 29%. So if you go for the high risk HPV DNA testing, HPV independent, HPV negative cancer will be missed entirely. I will come to the WHO classification just to let you give an idea. This is not a complete classification. This is the classification of the cervical cancer histology type. So, WHO in 2020 formally recognized HPV independent cervical cancer. Independent and HPV negative are different things. So, gastric type, clear cell type, mesonephric, endometrioid, neuroendocrine, carcinosarcoma, these cannot be detected at all by the high risk HPV DNA testing. Another interesting thing is that if you look at this chart, you will find that gastric type, mucinous adenocarcinoma, they are not preceded by CIN. And carcinosarcoma or sarcoma or melanoma, they can be only detected by the molecular profiling. No screening test detect these ever. So especially in women who is older than 35 to 40 years of their age, if you go for the high risk HPV DNA testing alone, you can miss these type, especially in a country like India, where the chances of having HPV negative or HPV independent cancers are very high. So this is the main reason. Financial is of course there, and then the co-testing, then the facilities there. So this is the 6 reasons high risk HPV DNA alone screening fails Indian women. 29% HPV negativity, which is 3 times more than the global rate. No Indian HPV type distribution data is available to us. In which particular state or which community, which type of the cancers are available, that data is not there. And our women, or they are anemic, they are malnourished. Girls have started their sexual activity quite early. They have got multiple sexual partners, and all this can lead to the HPV independent or HPV negative cervical cancer. And we don't have the infrastructure which can be scaled up. HPV screening guidelines are built on CIN2 or 3 reduction, not RCT proven cervical cancer mortality reduction. I hope you understand that doing high risk HPV DNA testing has not proved that it can reduce the cervical cancer mortality. So what's the fun in doing? It is better that we go for the FAP or LBC, and because it detects HPV independent cancers as well as HPV negative cancers also. Now coming a bit regarding the HPV vaccine, the evidence, the gaps, the adjuvant. So HPV vaccine randomized control trials, the endpoints is the problem. The basic problem is that no randomized control, in none of the randomized control trials, cervical cancer had been the endpoint. And it has been proved by Rice et al. That all 12 pivotal HPV vaccine RCTs were not designed to detect reduction in the invasive cervical cancer or mortality. After this, after 2020, two studies had come, one by the Lee, which has been quoted in a big way by all the oncologists and lot of obstetrician and gynecologists, even Dr. Nirja Bartla, that Lee study, because in that study, the endpoint was cervical cancer. But the main problem with that study was that it was studied till the age of 31, which is not the age for the cervical cancer. So that is between the vaccine and unvaccinated. Okay, and they have studied the vaccinated group till the age of 31 only. So on the basis of that, we cannot say that the vaccine can prevent the cervical cancer. Another important things which you have to, it's not mandatory that you understand, but you should know, because I have spent a lot of time in understanding and reading, and initially I didn't knew at all. That is why I repeatedly say that we need not give the testing, we need not give the vaccines, because there's so much problems with the trials, and there's as you have not gone into the depths, you don't know what is the seropositivity, what are the tests which have been done, you don't know about the immunobridging. So, and moreover, the basic test done was ELISA, and it has to be done along with the PBNA testing. I don't think any one of you must be knowing what is the PBNA testing. So that with all these flaws in the randomized control trials, that is why I just say there's no point in giving the vaccine. Now, a bit about these, that what the trials use as the primary endpoints. One is the seronegativity antibody titers. This is not a clinical endpoint and does not equal protection against cancer. So they have used the seropositivity or seronegativity and antibody titer, which are not the clinical endpoint. Then they have measured CIN2 or CIN3 lesion reduction, but it is a surrogate endpoint. As I told you, most of the trials, they never measured the endpoint as a cervical cancer as an endpoint. They have basically measured CIN2, CIN3 lesion reduction, which is a surrogate endpoint. And CIN2 and CIN3, we very well know that at least CIN2, 60 to 70% lesions regress spontaneously without treatment, and 30 to 40% of the CIN3, they also regress without treatment. Then they have measured persistent infection reduction, which is also a surrogate point, which is not an endpoint for the cervical cancer. Cytological testing in trials may have captured self-regressing lesions. So this is very important. That is why I say that HPV vaccine is not needed. Then another important factor, which most of the gynecologists are not aware, that in every trial we know this thing, that we have to do the placebo or the controls. The control has to be given the saline, but in all the Gardasil trials they have given the aluminium, and especially in the Gardasil they have used a novel adjuvant which is AAHS. Only 700 patients in Gardasil 4 trial they have given the normal saline to the control group, and otherwise in all they have compared vaccinated versus AHS, they were given AAHS adjuvants. So the aluminium, now it is well known, can cause multiple problems, especially it gets deposited in the brain, later on leading to the Alzheimer, Parkinsonism, and many other diseases. So this is one of the reason, and the problems arising because of the adjuvants, they have been suppressed by the pharma. So the control group problem is there. It is a novel adjuvant. It has got a unique risk profile which has not been studied. It has got long-term systemic effects of AHS is unknown, and Gardasil 9 has got the higher AHS load, and we have got there is a regulatory caveat, that is FDA AMA accepted safety based on AHS control data and incomplete evidence based by design. Both regulators mandate ongoing post-marketing surveillance, which is not occurring. And it is not, as I told you just now, it is not equivalent to standard placebo. So now coming to the HPV vaccine adverse events post-licensure record. So what is happening in the post-licensure signals under investigation or debated, and which have been suppressed by the pharma company, for which I am repeatedly telling, is that POTS, that is postural orthostatic tachycardia syndrome, multiple cases clustered globally, mechanistic plausibility via autoimmune pathway, that is CRPS, that is complex regional pain syndrome. Japan suspended government recommendation in 2013 due to CRPS spots cluster, partially resumed. It has resumed because of the, again, I will say it is pharma, under the pharma company. Premature ovarian insufficiency, case reports linked to Gardasil, mechanism proposed via AHS, autoimmune cross-reactivity, autoimmune condition like SLE, MS-like illness, Guillain-Barré syndrome, various reports, association not yet casually established in RCT. Denmark EMA reviewed around between 2015 to 2016 more than 1600 CRPS reports across EU following Gardasil. Conclusion, but the conclusion drawn is casualty not proven but not excluded. So because of this again reason, there's a suppressed data concern. The future trial data included AHS controlled comparison. Since adverse events were benchmarked against an active comparator, AHS not saline, true placebo reference adverse events rates have never been independently published. So we are not supposed to destroy the future of our young girls. Long-term, another important is that long-term efficacy, the unresolved question. See, in all the future two Costa Rica trials, 50% of the women or the girls, they had only, after the 10 or 12 years, 50% only had the positive antibodies. And again, it is not the seropositivity. The test done for ELISA, as I told you earlier, it has to be complemented with the pBNA testing. Seropositivity is not equivalent to neutralizing antibodies. That is very important. So what remains unproven or unknown? No RCT has measured reduction in invasive cervical cancer as primary endpoint. Antibody waning is a real challenge and problem. Whether a booster dose will be required is unknown. No long-term protocol exists. The lifetime protection from 2 doses. In India, we are giving only 1 dose is based on modeling, not 50 years follow-up data. So these are very important points, friends, which we should remember, because it's not about vaccines are not a panacea that we give to everyone and we will we'll reduce the cervical cancer. In fact, in coming years, in next 20 or 30 years, we will be seeing lot of morbidity and mortality. We are already seeing the morbidity and mortality because of the COVID vaccine. Protection in women with prior HPV exposure is substantially lower. We know this very well. And AHS long-term systemic effects remain uncharacterized. No independent 20-year safety follow-up for this proprietary adjuvant exists. And then type replacement is a major issue. What they have found that if suppose you give the Gardasil 4 or Gardasil 9, then the other type which are being not covered by the vaccine, they will start multiplying and they can cause a cervical cancer. So vaccination does not replace screening. That is also another important thing. So that is why I place lot of focus on the screening, and that too by the pap smear or LBC, followed by the, if needed, for the colposcopy. And that is why I'm repeatedly saying high risk HPV DNA, because HPV independent and HPV negative cancers are the major concern. So this is a HPV associated versus HPV independent cancer's clinical profile. Okay, so for many of the cancers, you require, for example, in the HPV independent cancer, you require different molecular profiling like P53, KRAS, PIK, 3C HPV independent cancer. I don't want to tell, but I need to tell that conflict of interest in the evidence-based in vaccine exists. WHO is being funded by Bill Gates, and all I have clearly found out that those who have recommended high-risk HPV DNA testing, those who have recommended 90, 70, 90, they have been all funded by Bill Gates, and it's a circular funding program. They have designed, Bill Gates funded WHO, IARC trials, and a lot of other trials, and they being they have the same people, they formed the group and became the advocates for the high risk HPV and DNA testing. They became the advocates for the vaccination. And it is very well documented that the diagnostic industry, Roche, Hologic, Kaizen, Abbott have directly funded landmark HPV screening trials. The COMPASS trial, Australia, foundational for national HPV screening policy, was co-funded by Roche Molecular System, the manufacturer of COBAS HPV test in the trial. It is the evidence-based recommendation for the Indian gynecologist. Insist on PAP LBC as minimum standard. Never dismiss symptomatic women on negative high-risk HPV testing. Apply cofactor informed risk assessment at every consultation. Document HIV status, tobacco, OCP duration, parity, nutritional status, co-infection, multiple sexual partners. Heightened vigilance for women over 40 and postmenopausal. Counsel completely on the vaccine limitations. Vaccinated women must continue the screening. Refer unusual adenocarcinoma for specialist review, and advocate district level screening infrastructure. This is very important. All of us, those who are working at the district level, they need to write to the Ministry of Health and Welfare that what are the facilities being needed. If the government of India is trying to reduce or eliminate cervical cancer completely without proper labs, without proper assistance to us, to the gynecologists in the form of colposcope or in the form of the laboratory, we cannot eliminate. Thank you.